Welcome back, everyone. This is the last chapter of the second section of your textbook, so it generally wraps up the information that your textbook provides on the biochemical basis of LTP and learning and memory. I can hear a collective sigh from some of you who may have broad interests in the neurobiology of learning and memory, but perhaps not so much interest in the molecular and biochemical basis of learning. After all, behavioral neuroscience is a broad discipline that uses many approaches to investigate the biological basis of behavior, ranging from the cellular and biochemical basis to the systems levels of analysis that we talked about in the first lecture for Chapter 2. So if you're in that camp, well, rejoice, because after this chapter, we'll move on to more circuit and system levels of analysis. We will occasionally return to talk about some biochemical supports for these circuit and systems modifications, like visiting with our old friend Krebs in some interesting experiments. But we have, for the most part, now learned the fundamental mechanisms that encode molecular and cellular structural changes in potentiated neurons. In fact, Chapter 8 in your textbook consists simply of a summary of the foregoing 7 chapters. It doesn't really go into any new information whatsoever. So I'm going to play along and provide a brief summary in line with the content of Chapter 8. However, I also want to take advantage of the time that we have available, since Chapter 8 is not such a voluminous read, and tell you about another experiment that I think will nicely bridge our review of cellular and molecular mechanisms in LTP to more circuit levels of analysis of the biological basis of learning. I'm going to tell you about a really amazing experiment that links LTP and learning. I think it's actually a very compelling story. In fact, I often joke that trying to find the synapses or cells that underlie the learning of an event or a skill would be like trying to find a needle in a haystack. You got 86 billion neurons in your brain. Where are you going to find that potentiated synapse? However, at the risk of revealing too much about the experiment again before we get into it, I do feel that they put together a really smart experiment to, in a way, well, almost find that elusive needle. But we'll get to that in a few minutes after I do Dr. Rudy's summary of chapters 1 to 7. Oh, and I know before I even, hardly start to record this lecture, that it will be fairly short. But I'm going to be posting it for Friday of the third week of the course, and I am quite certain that everyone will appreciate having a little bit of a break on a Friday. So here goes. In the summary of chapters 1 to 7 that's offered in chapter 8 of your textbook, there's a diagram with a proposal for 4 stages of LTP, which I've copied onto this slide. Now, I've been referring to these 4 stages loosely as induction or short-term LTP, the early phase of late LTP, the later phase of late LTP, and maintenance of LTP. And you'll find similar listings of the stages in many sources that describe the organization of LTP across time. But you will also find sources that describe it more in line with the description in Chapter 8 of your textbook. And I do actually like this taxonomy that was offered up by Dr. Rudy in Chapter 8. So the first stage of LTP is known as generation or induction. And in this stage, within about a minute after the application of tetanic stimulation, NMDA receptors allow in sufficient calcium to activate kinases that phosphorylate mechanisms that accomplish a variety of potentiating functions in the spine. These mechanisms disassemble actin at the head of the spine, insert GLU-A homotetrameric AMPA receptors into the membrane, translocate these newly inserted receptors to the active zone of the postsynaptic membrane, and temporarily trap these new receptors into the membrane, and also activate the UPS system to degrade excess signaling proteins, thereby limiting the overall magnitude of the potentiation of the spine. The second stage is called stabilization. It starts around 15 or 20 minutes roughly after tetanic stimulation, if we use standard induction protocols in, let's say, a Schaefer collateral to CA1 pyramidal cell preparation. Now, similar timeframes are generally reported for other preparations as well. But in this 15 or 20 minute time frame, calcium levels continue to rise as calcium-induced calcium release is triggered by the NMDA receptor mediated calcium influx and by the actions of BDNF, overall impacting ryanodine receptors in the endoplasmic reticulum of the spine. And then the metabotropic glutamate receptors, the mGluR1s, they mediate increases in inositol triphosphate signaling that activates calcium release through IP3 receptors in the endoplasmic reticulum of the dendrite and somewhat into the spine. The stage 2 box in this figure from your textbook summary doesn't highlight this, but these actions also initiate local translation of constitutively available mRNA that support the continued provision of signaling molecules like kinases and AMPA receptors. Now, this does appear to be recognized by the vertical brown box that shows which stages involve biosynthetic actions. There's also movement of actin that causes the neck to retract and the spine head to start to enlarge. The third stage is consolidation, which requires from 2 to 4 hours to initiate in a standard preparation. This is the stage that is supported by genomic cascades of signaling. BDNF and mGluR activity continues to hold plasticity, to hold postsynaptic calcium concentrations at an elevated peak. But then signals arrive at the nucleus, and genomic transcription of plasticity products begins to provide specific mRNA and proteins that are targeted in large granules out to the dendrites to be captured and used at the activated spines. The spines that were tagged by the stimulation are capable of capturing relevant proteins, and they have a fully activated cascade of mTOR signaling now to promote translation of top mRNAs. In other words, they are fully engaged in translating the relevant mRNA plasticity products from the genomic signaling. Furthermore, during the consolidation stage, the UPS system is engaged in degrading repressor proteins that would otherwise limit and perhaps slow down the genomic cascades that are responsible for the consolidation of the potentiation of stimulated spines. And of course, the last stage is maintenance over the long term that could range from days to weeks to even years or a lifetime. And in the intact animal, this is mediated by protein kinase M-zeta, or PKM-zeta. Of course, the function of this memory molecule is essential for maintaining the ongoing and persistent presence of phosphorylated GLU-A2 containing heterotetramers in the postsynaptic membrane, by supporting the exocytosis and insertion of these receptors and reducing the endocytosis and removal of these receptors in the face of constant molecular turnover. And lastly, let's address the vertical bars at the sides of the figure, including the brown one that I mentioned a few minutes ago. But I'll start with the blue vertical box that points out that post-translational modifications are involved in all 4 stages of LTP. And of course, then by inference, all 4 are implicated in the neurobiology of learning and memory in the real world. Of course, in stage 1, post-translational processing, mostly kinase-mediated phosphorylation, is pretty much the central event. And there's no evidence of significant transcriptional or translational activities at this stage. And clearly, post-translational processing activities continue on through all of the rest of the stages, even including lifelong maintenance, as we have to keep adjusting to compensate for molecular turnover to replace GluA2 heterotetramers and to keep the potential to respond to input at an elevated level. The vertical brown bar on the left points out that transcription and translation start around the 2nd stage, although I would argue that even if genomic signaling to increase specific transcriptional activities may start in stage 2, it's not extensive enough during this stage to promote any functional consequences just yet. And indeed, translational activity must endure even through the 4th long-term maintenance stage, because protein kinase M-zeta is a self-replicating, constitutively active signal at potentiated spines that promotes its own translation from mRNA to keep the potentiation going. So that covers the summary offered in Chapter 8 of the textbook. Now let's do the bridge. And I'm going to talk to you about a set of very exciting experiments done by Rio Peddotti and collaborators, exploring a link between LTP and circuits that mediate learning of behaviors. I'm going to start the story of this experiment by talking about handedness. We all know that some people are right-handed and some people are left-handed. And of course, this doesn't mean that they cannot use the contralateral limb. It just means that they have a preferred and more skilled hand and arm for engaging in motor tasks. And we also know that the left somatosensory cortex receives input from the right hand and arm, and the left motor cortex controls the right hand and arm, and vice versa, at least for the most part. There is some crossover. Well, these basic biological characteristics are very well documented in humans. So would it surprise you to know that the same is kind of true for rats? Each rat seems to have a preferred forelimb that it uses for reaching and grasping, and it's controlled by the contralateral hemisphere. Now, I don't think their degree of lateralization is quite as extensive as ours, and they don't prefer one forelimb as much as most of us do, probably mostly because they don't have the specialized skilled control of the hand, or in this case the paw, like we do. However, if you place a rat into an experimental task situation where the animal has to reach and grasp with a forepaw, they will reliably use one preferred forepaw over the other. So that's what Rio Peddotti and collaborators did. They placed hungry rats into an acrylic arena with a narrow slot at one end, shown right here. They then placed a nugget of food at the limit of the rat's reach so that it had to extend its paw through the narrow slot, pick up the tasty morsel, and bring it back through the narrow slot to eat it without dropping it. Now, for a rat, that's an extremely challenging task, testing the limit of its potential to learn a fine motor skill. But note that it is a skill a rat can learn. My dog, for example, is a very intelligent creature by comparison, but he can't manage to pick up any small objects with his forepaws. He can merely bat things about as he doesn't have dexterous little hands. But rats are actually quite capable of manipulating objects with their forepaws, as you can see in the picture. They're indeed actually quite dexterous. So, Riupodati trained the rats for 5 days and found that the rats' abilities reached asymptotic performance after about 3 days. The dependent measure in this experiment was a ratio of the amount of food consumed over the total amount of food available. So the food pellets were carefully weighed before the experiment. The rat might successfully consume some of it and drop some of it where it would fall down into a tray below the arena. So the experimenters simply subtract the spillage from the amount of food offered, and they have a measure of the success of the animal at consuming the pasta. Okay, I'm going to take a brief time out to tell you about something that I find objectionable in this experiment. If you look at the learning curve on the graph, they report how many rats were tested on each day of the experiment. Now that's good, but what you can see here is there's a sudden drop from 95 animals on days 1 and 2 to 57 animals on day 3 and a few less still on days 4 and 5. Now, the authors of this research report that was published in the journal Science, they don't explain why the numbers dropped, but suddenly the curve flattened on days 3 to 5. Did they perhaps eliminate all animals that were unable to perform the task? I don't actually think that would be an unreasonable thing to do, as any rats that can't learn would provide meaningless distortions in the data. Or perhaps did they drop all the animals from the experiment that didn't actually use a preferred forepaw and rather switched back and forth between paws? That would also distort the outcome of the experiment, which I think you'll appreciate better after I show you a little bit more of the methodology. So, I don't think that what they did was particularly unreasonable, as they kept the rats that had learned the task and did it at a high level of performance, up to about 90% efficiency, using a preferred forepaw. However, it's uncomfortable that they were not fully forthcoming in their data reporting. We have a large push in neuroscience to engage in transparent reporting of our research. So a word to the wise: don't do this. When you report data from the experiments you conduct, please be fully transparent in your reporting. Okay, so now that I've shown you the basic structure of the behavioral methodology, and I've stated my critique of their presentation of their experiment, let's actually return to the experiment. The rats were trained on the task until they reached asymptotic performance, which means that the rats that were used in the experiment reached a level of performance that indicates that they had maxed out their ability to actually learn the task. So when this high level of skill was obtained on the 5th day, the brains were extracted from the rats and sectioned in the coronal plane. Bilateral recording electrodes, shown here and here, were placed in the forelimb region of the primary motor cortex. And stimulating electrodes were positioned at inputs to those regions. The electrodes were positioned over the cortex contralateral to the preferred limb, so this is referred to as trained cortex, and over the cortex ipsilateral to the preferred forelimb, so this is designated as untrained cortex. Now they also implanted electrodes over motor cortex representing hind limb function for the contralateral and ipsilateral hind limb. And in addition, they had some paired control sections from animals that did not undergo training. Now next, they administered single pulses of depolarizing current through the stimulating electrodes. Now note that this is not tetanic stimulation, just single test pulses. And they recorded the field potential response of the motor cortex through the associated recording electrode. They found that the field potential response for stimulation of the trained forelimb cortex was greater than the response for stimulation of the untrained cortex. In other words, the synapses that processed motor function in the cortex that represented the preferred limb that was trained to pick up the pellets was potentiated. The strength of these synapses appeared to be greater than the strength of synapses in the motor cortex representing the forelimb that the rats did not use in the reaching and grasping task. And of course, examination of the hind limb cortex and the forelimb cortex of the paired controls revealed no differences between the hemispheres in strength of signaling. So do you see what I mean? They've almost found a needle in a haystack. By doing a rigorous training in a highly skilled task for a rat, they were able to identify a circuit in the brain in which synaptic strength appeared to be increased. Hypothetically, that increase in field strength represents the learning. Okay, but I don't think they were totally convinced yet, and frankly, neither am I. Although I don't believe this is an adequate explanation for their findings, a plausible explanation is simply that the stimulating and recording electrodes just happen to be better aligned in the trained forelimb cortex than they were in the untrained forelimb cortex. Because a very small misalignment of the electrodes could have a significant impact on the magnitude of the response to stimulation. But never fear, Riu Pedotti was not done yet. The next set of experiments in their science paper, I think, is super cool. In this experiment, another cohort of rats was trained for 5 days in the forelimb reaching and grasping task until they reached that high level of proficiency. And stimulating and recording electrode pairs were placed once again in forelimb motor cortex for contralateral trained cortex and ipsilateral untrained cortex. But now, rather than just recording responses to test pulses, they induced LTP in the motor cortex. In the top graph, you can see what happened when they applied tetanic stimulation in the trained cortex. Tetanic stimulation right here. The stimulation did not produce LTP in this rat. Then they even did it a second time, and it still did not produce potentiation. Okay, let's look at the lower graph for just a minute and see what happened when they applied tetanic stimulation to the untrained cortex. The first stimulation, right here, using the exact same stimulation parameters as they used in the trained cortex, produced a nice LTP. And the second stimulation, here, produced a slight enhancement of the LTP. However, a third stimulation and a fourth tetanic stimulation did not enhance the LTP any further. It seemed that after the first 2 rounds of tetanic stimulation, the synapses were fundamentally saturated and could not be potentiated any further. So do you see where I'm going with this? It appears that in the trained motor cortex, up here, the synapses were fully potentiated by the training even before the first tetanic stimulation. So they couldn't be potentiated any further. They were saturated by the training. In contrast, the untrained cortex responded to 2 rounds of tetanic stimulation, and it was only after 2 rounds of stimulation that the synapses were maxed out. Now, let's look at the upper graph for the trained cortex again and follow through with some of the manipulations that I didn't finish on that graph. So the first 2 times they stimulated and they got no potentiation. So the third time they didn't provide tetanic stimulation. Rather, they applied weak, low-frequency stimulation. And the cortical responses showed a nice LTD. Now, this reveals that the synapses were capable of plasticity. They just weren't capable of potentiation. The failure to induce potentiation was then not because of any misalignment of the electrodes, because they responded with the LTD induction. So everything seems to be working, but the synapses couldn't be potentiated. Excuse me. So next, excuse me. So next they renormalized the baseline, meaning they took this baseline and then just kind of set it at the 100% baseline mark, just to line up everything again. And then they applied tetanic stimulation. Now the synapses actually potentiated. In other words, the synaptic strength in the trained motor cortex was initially saturated. The synapses were maximally potentiated. And then application of weak, low-frequency stimulation lowered the synaptic strength, so a subsequent round of tetanic stimulation could potentiate it again. Not only had Ruo Padodi found a needle in a haystack and made a connection between a learning task and potentiation in a specific circuit, but it also seems that they had uncovered an opportunity to explore the range of potentiation that can be expressed in the motor cortex neurons. And we're going to see that in just a minute. I want you to note that the data reported on the graphs on the previous slide represented responding to the electrophysiological manipulations in a single rat cortex. And in fact, it was a thoroughly rare case in which the behavioral training resulted in a complete saturation or a complete inability to induce any potentiation. So the graphs on this slide represent the means from all the animals. In this case, you can see that tetanic stimulation did produce a small LTP in the trained cortex, where the synaptic strength on average rose to only 114% of the baseline, and the maximum was reached after only 1.18 trains of stimulation on average, meaning that it took only one train of stimulation to reach asymptote in most of the rat cortices, and it took 2 trains only in a very few, averaging out at 1.18. And actually 3 of the 11 rats tested responded like the rat in the previous slide, where stimulation produced no potentiation whatsoever. And on the other hand, tetanic stimulation of the untrained cortex increased the synaptic strength to 152% of baseline, and it took an average of 2.9 trains of stimulation to get there. This makes it seem like the untrained cortex was much more potentiable. Could it be that the trained cortex had high synaptic strength, and the untrained cortex had low synaptic strength? So the trained cortex was near its max already, and the untrained cortex was far below its max for synaptic strength prior to the electrical stimulation. And it would seem that the answer to that question is yes. In their science paper, they reported another experiment in which they gave another cohort of rats 5 days of training. But this time, rather than applying tetanic stimulation, they administered weak low-frequency stimulation. And the results are shown on this slide. I want you to see that the trained cortex could be massively depressed, shown right here. And the untrained cortex, not so much. In fact, the trained cortex, which we hypothesized was potentiated to near its maximum strength by the end of the training, was depressed to 49% of that basal strength using 2.5 rounds of weak low-frequency stimulation on average. In contrast, the untrained cortex could only be reduced to 79% of its basal strength, and it reached this low after only 1.2 rounds of weak low-frequency stimulation. So it looks like the trained cortex was lowered down from on high, and the untrained cortex started low, so could only be moderately impacted by the depotentiation. And this, I think, is an awesome summary and conclusion drawn from the data by Rio Pedati et al. They proposed that a synapse can be modified within a limited set range. In the untrained control cortex on the left, you can see that the synaptic strength, measured by the field potential strength, is moderate in this naive preparation. It can be increased by a few rounds of tetanic stimulation until it reaches a maximum where additional rounds of tetanic stimulation would not increase it any further. Or it can be decreased by a couple of rounds of weak low-frequency stimulation to a minimum, where additional rounds of weak low-frequency stimulation would not suppress it further. But then let's look at what is happening with the trained cortex on the right. The model at the far right suggests that the range of modifiability could go up or down from any set value. So whatever the state of the synapses is at time 1, you could potentiate up a bit or down a bit. Well, this model in my mind doesn't make any sense and certainly is not supported by the data. Because in trained cortex, very little potentiation could be induced and a lot of depression could be induced. You see that the responses to test pulses in the graph fall below the range of potentiability after weak low frequency stimulation, and they do not reach the heights predicted by the upward shift in the range after tetanic stimulation in this model. So you know what? Let's just cross this model out. And let's look at the other model for the trained hemisphere instead. So this model kind of in the middle here, the model in the middle, shows that the range of modifiability is actually set. It's the same range right here as in the naive synapse, except we're starting out higher up in the range. So it's the same range that was seen in the model at the left for the naive preparation. In this model, we can potentiate a little bit and get to the maximum of the range. Or we can depress a lot and get to the minimum of the range. And so this model does fit with the data. So Rio Peddotti proposed that the range of modifiability is set and that we can reach the peak or the nadir. But it's within that range that we will be confined in terms of synaptic modifications. Now, I want to throw another idea out to you. I wonder if this model of range of potentiation might have implications for other learning situations we might consider. So, for example, in the first chapter, I defined learning as an enduring generation or modification of internal representations. And after a little discussion, concluded that the definition of learning should include sensitization to drugs of abuse and the process of addiction. So what if we could identify a set of connections that might be modified by drug exposure, like glutamatergic inputs to nucleus accumbens or inputs to the amygdala? Then expose the animals to the drug treatment or even self-administration of a drug like cocaine, and then see if it alters the magnitude of potentiability and the number of times, the number of trains of stimulation needed to reach maximum strength in comparison with, let's say, vehicle-treated controls. So I'm just thinking out loud here, but I think there could be a whole wealth of stimulating, and yes, pun intended, experiments that could be done by an enterprising young scientist, simply by adding this model to other issues we explore in plasticity in modern neuroscience. Anyway, that brings us to the end of this section of the textbook and the end of this chapter. And I'm just going to wish everybody a wonderful and safe weekend. And I will see you, or, well, not really, but I'll be here next Monday with Chapter 9. Have a good weekend.